We start in 5 minutes, so have a little patience and wait 5 more minutes so everyone has the chance to join, and see you in 5 more minutes. Okay, I think we can start. Hello and welcome everybody to our permeability series with the topic drug delivery to the brain. In situ gelling formulation enhances carbamazepine diffusing through nasal mucosa models with mucin. My name is Yannick and I will be your host for this webinar. We are happy to have our external speaker with Elisa Corazza the second time here at the Permeability series. She's a PhD candidate in the group of Professor Barbara Luppi from the University of Bologna in Italy. Her aim of the PhD project is to develop innovative strategies to improve local and/or systemic treatments by increasing drug penetration and permeation through the nasal mucosa. Moreover, part of the research considers the impact of probiotics and the metabolites on drug absorption. In her presentation, she will focus on the experimental setup with the preparation of the barrier and how you can use it for the nasal delivery. Elisa's research centers on optimizing a thermosensitive in situ gelling formulation, elevating intranasal and nose-to-brain delivery of the anti-epileptic drug carbamazepine, explore preliminary findings and innovative approaches that hold the potential to reshape drug delivery to the brain. If you have any questions, please feel free to type them in our Q&A window, and we will answer them at the end of the presentation. Without further ado, I give the word to Elisa. Enjoy the presentation and see you at the Q&A session again.
Okay, I'll start sharing my screen. Should start. Can you see my presentation?
Not yet.
Okay. Okay. Now you should.
Yes, we can see it.
Fine. So thank you, Yannick, for this kind introduction. And thank you for inviting me this second time to the PermeabilitySeries webinar. Today, I would like to share with you the result of our research into the use of in situ gelling formulation for the nose-to-brain delivery of carbamazepine. During my talk, I'll briefly introduce the main topic of this research, as well as the aim of the study. I will continue with the experimental section, and I will conclude by summarizing the result we obtained. As you may know, nasal drug delivery presents some undeniable advantageous features over other conventional routes of administration, namely improved compliance because of the avoidance of needle and the possibility of self-medication, the fast onset of action due to the relatively high permeability of the nasal mucosa, minor prosystemic metabolism, and most notably, this possibility of a direct access to the cerebrospinal fluid. And is in virtue of this latter point that nasal drug delivery is gaining attention as an alternative strategy to deliver drug whose target is located in the brain. However, as any other route of administration, it also presents some shortcomings, which are mainly related to the mucociliary clearance, the restricted nasal tissue surface area, and the barrier effect that is posed by both the nasal architecture and composition of the epithelium and the mucus gel layer that lies on it. So from this limit comes our commitment to investigate innovative strategies capable of improving drug absorption across this biological barrier. And in this context, a thermosensitive gelling system seems to be a promising strategy to address the downsides of this delivery route. These in situ gelling systems are characterized by being in solution at room temperature and are able to undergo a salt to gel transition due to hydrophobic interaction that occur between the components of the gel, and these interactions are triggered by an increase in body temperature. In virtue of how they behave, thermosensitive gelling system can adapt to the nasal cavity, are able to improve both the drug retention time at the administration site and its bioavailability, and they can also provide a controlled and sustained release of the loaded drug. Usually, thermosensitive systems that are intended for nasal delivery are made of poloxamer, in particular poloxamer P407 and P188. These poloxamer are synthetic 3-block copolymers They are characterized by good water solubility, are regarded to be neither toxic nor irritant for the mucosa, and they can also create, self-aggregate, better to say, into micelles. However, the gels that result from the mixing of these two poloxamer usually are characterized by weak properties in terms of mechanical strength and mucoadhesiveness. Therefore, they are usually blended with other polymers, such as bioadhesive polymers, included cellulose derivatives. In this study, we concentrate on hydroxypropyl methyl cellulose of two types, one high viscous, K100, and one low viscous, named here Metocel E50LV. The model drug of this study is carbamazepine. Carbamazepine represents the standard of care in the treatment of epilepsy and is usually administered orally with all the limitation posed by both the pharmacokinetic property of the drug and the route of administration. In fact, carbamazepine is characterized by a low water solubility but high permeability, and its low water solubility causes variable and low absorption across biological barriers. And at the same time, carbamazepine is subjected to relevant hepatic metabolism with the production of metabolites with a relevant clinical toxicity. Therefore, in an attempt of achieving a more effective strategy to treat epilepsy, we asked whether it was possible to rethink carbamazepine delivery through the nasal route. And to answer this question, we formulated some temperature trigger in situ gels based on poloxamer in the presence or absence of mucoadhesive polymers, and we characterized the resulting formulation for their gelling and mucoadhesive properties, for their ability to ensure a sustained release of the loaded drug, and to ensure permeation of CBZ across two different permapad-based models of the nasal mucosa. First of all, it was necessary to identify the best concentration ratio of the two poloxamer to obtain in situ gelling formulation capable of undergoing salt-to-gel transition at the physiological temperature of the nasal cavity. Therefore, at the very beginning, we simply prepared plain formulation, that is to say, made of the sole poloxamers, according to the cold method. Whereas when mucoadhesive formulation were prepared, the mucoadhesive polymers, so HPMC of the two types, were solubilized at room temperature in water prior to the addition of poloxamer. And the resulting formulation were at first characterized for their pH, temperature of gelation, and time to gelation, with the latter two parameters being measured according to the magnetic steering method. That is to say, determine the temperature or the time required for the magnetic steering bar to stop rotating as a result of the phase transition. From the first screening procedure, we were able to identify three formulations, in particular those characterized by 22% of P407 and from 3 to 5% of P188. And why we selected just these three? Because they were found suitable for nasal delivery. In fact, they exhibited a temperature of gelation comprised between 32 and 35 degrees, which is the physiological temperature of the nasal cavity, a time to gelation less than 60 seconds, which is necessary to avoid the anterior and posterior leakage from the nasal cavity, and a pH comprised between 5.5 and 6.5, which is the pH, the physiological pH of the nasal cavity. To these three formulations, we then added the mucoadhesive polymer, and the resulting mucoadhesive formulation were then characterized for the same parameters. And from this second screening procedure, we were able to select those formulations containing 4 and 5% of P188. And these mucoadhesive formulations, together with their plain counterparts, were selected to undergo further studies. Included the characterization of their mucoadhesive properties. First of all, we investigated their ability to influence on the time of mucadation. And time of mucadation was measured as the time required for the gel formulation to be completely removed from a specimen of sheep nasal mucosa while being subjected to a continuous flush of phosphate buffer pH 5.5, which basically was employed to mimic the mucociliary clearance. Results are summarized in this graph on the right side of the slide. And what we can observe is that when HPMC high viscosity, so K100, was added to the plain formulation made of 22% P407 and 4% P188, we were unable to detect a significant increase in the time of mucadation. Whereas when the same concentration of HPMC high viscosity was added to the plain formulation containing 22% P407 and 5% P188, the time of mucadation increased while increasing the concentration of the mucoadhesive polymer. However, an even more noticeable increase in the time of mucadation was observed when HPMC low viscosity was tested at its maximum concentration of 0.8% in the same plain formulation. And this last vehicle reached almost 30 minutes of adhesion. And of course, it was selected as the candidate mucoadhesive vehicle, which underwent further characterization step together with its plain counterpart. To get a better insight in the way E50LV was able to increase the mucoadhesiveness character of the nasal formulation, we measure the strength of mucadation. As you may know, the retention time of a formulation is also affected by the strength of interaction between the formulation and the mucosa. In fact, as this interaction becomes stronger and stronger, of course, the probability that the gelling system is longer retained at the administration site increase. Here we measure the strength of mucadation as the detachment force that was required to separate, to take apart a specimen of sheep nasal mucosa from the gel formulation. And here, once more, we demonstrated the role played by E50LV, as the force of mucadation was significantly higher in the mucoadhesive formulation compared to its plain counterpart. As I said at the beginning, poloxamer are able to self-aggregate into micelle, which can be of extreme importance when you have a low water-soluble drug. So in this study, what we did was also to test this gelling system ability to act as solubilizing agents. Therefore, we dissolved increasing concentration of CBZ from 0.25 milligram per milliliter up to 2 milligram per milliliter in the candidate mucoadhesive vehicle and its plain counterpart. And the resulting samples were first analyzed for their turbidity at 650 nanometers. As we can see in figure A, we can observe that for concentrations up to 1 milligram per milliliter, no turbidity raise was observed. Whereas when CBZ concentration was increased up to 2 milligram per milliliter, then a turbidity increase was observed, meaning that no more drug molecules could be solubilized. However, what we can observe is also that it seems that the mucoadhesive formulation exhibit a superior ability to solubilize CBZ. To further confirm these first results, the same samples were centrifuged to remove the excess amount of CBZ and were analyzed for their absorbance at 286 nanometers, which is the absorption peak of CBZ. If we look at the plain formulation, we see that there's an increase in absorbance for samples up to 1 milligram per milliliter. After this concentration, we see there's the value of absorbance remain constant, supporting our first hypothesis that no more drug molecules could be solubilized. But when the CBZ was dissolved in the mucoadhesive formulation, a continuously growing trend was observed, supporting the hypothesis of his superior ability to solubilize the drug. Overall, we can state that both formulation enabled an increased solubilization of CBZ, because CBZ water solubility was quantified to be around 0.118 milligram per milliliter, and that at 1 milligram per milliliter, we were sure that all CBZ was completely solubilized. To exploit the gelling and mucoadhesive properties of this formulation, they must be easily and correctly instilled in the nasal cavity. And since the first idea was to deliver this vehicle through a nasal spray, it is suggestible to keep the viscosity of the formulation at room temperature below 500 millipascal per second. Therefore, we measure the viscosity of our formulation at 3 different temperatures, 20, 25, and 35 degrees. And as we can see from this table, for both formulations, we were able to detect a slight increase in viscosity from 20 to 25 degrees, but the values obtained were still lower than the threshold, meaning that our vehicles could be easily administered in the nasal cavity. Then going further to 35 degrees, of course, there was a greater increase in viscosity, which support the idea that the salt to gel transition took place. Another characterization was made on the upon drug loading. So what we wanted to check was the drug loading influence on gel properties. So the two formulations were loaded with a drug and at a concentration of 1 milligram per milliliter, and parameters such as pH, temperature to dilation, time to gelation, and the detachment force were measured again and compared to the values we obtained with the unloaded formulation. And as it comes clear from this graph, despite the presence of the drug, the candidate formulation maintained the desired features. One of the aims of this study was to develop formulations that were able to release the loaded drug in a controlled manner. So we performed drug release studies across the dialysis membrane cut off 6 to 8 kilodaltons, and we tested 3 different samples: our candidate mucoadhesive formulation and its plain counterpart when loaded at 1 milligram per milliliter of CBZ, compared to a control solution that was a saturated solution of CBZ in ultra-pure water. Of course, because of the way we prepared the control sample, the drug was immediately available for release. And in fact, after 3 hours, almost 100% of the drug was found released. Conversely, if we look at the release profile of the two in situ gelling formulation, we can observe a more prolonged and slower release profile. And after 5 hours, almost 50% of the loaded drug was found released. So overall, we can state that both the mucoadhesive formulation were able to provide a controlled and sustained release of the model drug. As I said at the beginning, the pharmaceutical target of CBZ is located in the brain, and the advantage of the nasal route is that this drug can reach the central nervous system through 3 different pathways: the systemic pathway, that is to say, through the blood circulation, the trigeminal pathway, that means through the trigeminal nerves, or the olfactory neurons, which are located in the olfactory nasal region. Despite the advantage of having multiple uptake routes, the drug has always to cross some physical barriers, because if we think at the systemic pathway, to reach the blood circulation, the drug must cross both the nasal epithelium and the mucus gel layer that lies on it. But similarly, if the olfactory or trigeminal nerve fiber pathway is preferred, part of this barrier remain, because neither the olfactory neuron nor the trigeminal nerve fibers are directly exposed to the nasal cavity. Therefore, we wanted to make sure that our in situ gelling formulation were able to ensure CBZ permeation across both these barriers. And we planned our drug permeation studies across two different PermaPad-based models. The first was characterized by the standard PermaPad, which was useful to mimic the composition of the nasal tissue, whereas the other one was made of the standard PermaPad functionalized with a reconstituted layer of mucin. So in this case, we were able to mimic both barriers, so the nasal epithelium together with the mucus layer. And we tested the same samples that were analyzed during the drug release study. The permeation profiles were used to determine the drug flux across the different barriers in the different conditions, and the results are summarized in this graph. Well, if we look just at the control, so the CBZ saturated solution, what catches the eye is the negative impact that mucin has on drug flux, because the diffusion of CBZ across the PermaPad barrier was remarkably decreased compared to the sole PermaPad in the presence of the mucin layer. Something similar happened also when the CBZ was loaded in the plain formulation. However, it must be highlighted that in this case, the drug flux was reduced to a lower extent in the presence of mucin compared to the control sample. But what was really surprising was that when CBZ was loaded in the mucadesic formulation, this dramatic effect of mucin wasn't observed. In fact, there were no significant difference between the sole PermaPad and the PermaPad with mucin. Therefore, we can say that the presence of HPMC low viscosity at a concentration of 0.8% in the plain formulation played a pivotal role. On the one hand, it was necessary to increase the mucodesiveness power of the in situ gelling formulation, and on the other hand, it ensured CBZ permeability across both the standard PermaPad and the PermaPad with the mucin layer. To conclude, the vehicle, the in situ gelling thermosensitive system that we developed, made of 22% P407, 5% P188, and 0.8% E50 LVU, showed all the properties required by an in situ gelling vehicle for being delivered through the nasal route. It was able to strengthen the interaction with the mucus layer, increasing the mucodesiveness properties of the vehicle made of the two poloxamers. It was able to provide a controlled release of CBZ, and it ensured CBZ permeation despite the presence of mucin. Before moving on to the Q&A section, let me thank all the people who contributed to this work. First of all, my colleagues from the Drug Delivery Lab at University of Bologna, Professor Annette Bauer-Brandl from the University of Southern Denmark, Professor Massimiliano Piodicagno from the University of Oslo, and of course, Fabio for inviting me, and all of you for your kind attention.
Thank you very much. Can you hear me?
Yes, I can.
Yeah, okay, very good. Thanks again for your nice presentation and showing your research to the community. I think it's a very nice topic because you also got the recognition of a paper of the year for 2022. And this is my personal question. Have you planned any further studies regarding this topic of formulation?
Yes, we are now trying to exploit the same in situ gelling system for the delivery of probiotic cells to the through the nasal route. I think we are going to plan also other in vitro studies through biomimetic membranes. So yes, it's currently under investigation. We are also planning, we already used the same in vitro model for studying other formulation always intended for nasal delivery, but not as an in situ gelling formulation, as a solution.
Okay, nice. And have you planned something with comparison to cell models or ex vivo tissue to have the comparison
yes,
there?
something that hopefully we are going to publish in a few weeks. And yes, we compared the PermePad model with a cell-based model, primary cell isolated from the olfactory nose of pigs, and another a tissue-based model. So the same, always an olfactory mucosa, always from pig, but directly the tissue excised from
Yeah, ex vivo. Yeah, okay. Yeah, okay. Very interesting. Looking forward to the results.
And we got very nice results, very promising for PermePad.
Thank you. And then another question regarding the application. How high was the mucin layer on top of it? Is there an explanation of why it's so decreased? Because last month we had the nice presentation from Cosmin. He used a mucus for the length, and he was able to show not always a decrease, but sometimes an increase as well for different compounds.
I mean, our choice was to add as much mucin as to cover completely the surface of the PermePad. So we didn't really wanted to have a thick layer, but as thick as to cover the surface. So we didn't really measure the thickness of this layer. I can tell you that for an area of almost 2 centimeters square, 200 microliters of this solution were necessary. So it's really just a very, very thin layer of mucin on the top. So we observe this effect without the necessity to create such a thick barrier. Also because it's not something that would represent what really is in vivo, because in the physiological mucus layer is just 5 micrometers in the nose. Mm-hmm. So it's very thin.
Very thin. Okay. Very good. Anyone else questions? Please feel free to type in your questions in the chat. I think Elisa is happy to answer them.
Yes, of course.
I saw some registrations from Asia, but of course it's during the night right now. Maybe they will arise after the recording.
Oh yes, but I'm available. If someone has questions, you can email me, and I'm really happy to answer you.
I type in the email address. You can contact us always, and we can forward the questions to Elisa. And I will now unlock or make it available to download the presentation from Elisa. So if no one else have a question, I think we're good for now. Please feel free to contact us if you have further questions in the future. Lisa, all the best for your research, and great Christmas. And
You too.
we see us again next year with the Permeability series, and I'm very happy
Thank you.
to continue this collaboration. Thank you very much.
You too. Bye.
Bye bye.